Today I'm going to be talking about GPCRs. GPCRs are G Protein-Coupled Receptors that are expressed on cell membranes to detect and transmit signals. I'm going to mostly be using this poster published by Cayman Chemical. You can find this poster on their website and you can get your own free copy. I'll put the link in the description. And I'm going to be using this poster in order to show you guys the pathways that GPCRs use to transmit signals. Okay, first up we have this image of a GPCR. We have the squiggly lines, which is the transmembrane part. It goes inside and then outside of a cell 7 times. And then on the inside we have this thing called a G protein. And the G protein has 3 subunits, an alpha, beta, and gamma subunit. Today I'm only going to be discussing the effects of the alpha subunit and the pathways that it takes in order to transmit a signal. The alpha subunit of a G protein can be broken down into 3 subtypes, the GS, the GI, and the GQ. And these subtypes use different pathways in order to transmit a signal inside the cell. Here we have a close-up of the GS and GI subtype pathways. This image was pulled from the poster that I showed you earlier. On the left side we have the GS subtype. The first thing that happens is a ligand binds to the transmembrane part of the GPCR. This induces a conformational change that allows the G protein to be released. The GS alpha subunit travels along the inside of the cell membrane to activate adenylate cyclase. Adenylate cyclase then converts ATP into cyclic AMP. Cyclic AMP is a secondary messenger. It causes a variety of effects downstream. A major effect to remember is the activation of protein kinase A. To remember the pathway of this subtype, keep in mind that S indicates stimulating, in this case the stimulation of adenylate cyclase. On the right side we have the GI subtype. As GS stimulates adenylate cyclase, GI inhibits the activity of adenylate cyclase, blocking the production of cyclic AMP, thus dampening signal transduction caused by molecules that activated GPCRs of the GS subtype. The GI subtype also has pathways of its own. These include the activation of PI3 kinase, which activates protein kinase B, also known as AKT. The GI subtype also activates the RAS protein, causing another signaling cascade involving MAPK. Moving on to the third major subtype of GPCRs, the GQ subtype. This subtype uses pathways independent of the GS and GI pathways. When a GPCR of this subtype is activated, the GQ subunit travels along the interior of the cell membrane to activate phospholipase C. Phospholipase C breaks a phospholipid from the cell membrane called PIP2 into 2 pieces. One piece we'll call DAG and the other piece IP3. IP3 is an intracellular secondary messenger that activates calcium channels on the membrane of the endoplasmic reticulum. This allows stored calcium to be released. DAG activates calcium channels on the membrane of the cell itself, allowing calcium from outside of the cell to enter into the cell. DAG and calcium present together can activate protein kinase C. Protein kinase C transduces the original signal of the GPCR further downstream. Another thing to keep in mind is that calcium ions are also secondary messengers that can cause their own effects downstream. Now that we have discussed a little about the structure and mechanism of GPCRs, I'm going to explain why I decided to make a video about them in the first place. GPCRs regulate many physiological processes in the human body. They are crucial in maintaining homeostasis. Numerous neurotransmitters, including dopamine and GABA, use GPCRs to induce responses. They also play a role in tumor development and immune response. Because of their wide-ranging abilities, 1/3 of all FDA-approved drugs target these receptors. One of the main reasons for their approval rate is that GPCRs are highly specific. An indication of how specific they are is clear due to their use in the synaptic clefts of neurons. In this environment, different GPCRs can be positioned very close to one another and still induce unique responses upon activation. While researching the pathways of GPCRs, I stumbled across a YouTube video posted by Nano. In it, they are interviewing a psychedelic researcher discussing his findings on genetic mutations in these receptors and the effect those mutations had on dosage response. He found that a mutation resulting in the change of a single amino acid can have substantial effect on an individual's response to a prescribed dosage.
First figure, which is a snake plot showing all the mutated amino acids in pink. As you can see here, this is the extracellular domain. And down here, this is the intracellular domain, right? Which couples with downstream signaling proteins, right? Such as beta-arrestin and G protein. So what's interesting here, Asher? Can you comment on that?
Yeah, I mean, overall, what I want to talk about is the fact that there's a histidine residue down here, 452, that when it gets mutated, it actually has a pretty drastic effect on cellular signaling outcomes of a lot of the psychedelics. But then, if you go to the top a little bit, Daniel, near extracellular loop number 2, this residue mutation here, which is actually part of the lid mechanism that Brian Roth's research group discovered, it actually does not affect psychedelics too much, which is interesting because it's within the orthosteric binding site.
Right.
And now let's go down to some of the plots.
Okay, so let's discuss figure 2. We're talking about efficiency and potency here, right? We have psilocin and mescaline.
We're basically talking about the efficacy and the potency, about how when we have a histidine 452 mutation and then we have psilocin as our drug, look how much drastically less efficacious the compound is with that mutation. It's huge.
Right.
And also there's a change in the potency, clearly. And if we compare that, let's say, to just for example, serine 12 aspargine, notice how that mutation at mescaline, when you have the drug, the potency actually increases.
After watching that YouTube video, I found an article that wanted to study that phenomenon in detail titled Pharmacogenomics of GPCR Drug Targets. Pharmaco meaning it's related to drug action, and genomics meaning it's related to complete sets of DNA data. In the paper, they looked into how variable the genetic coding for G protein-coupled receptors are in a population. Are individuals with these varied genetics likely to respond differently to drugs? And how much is this costing the pharmaceutical industry, since their drugs may not be performing as expected? GPCR targeting drugs account for over $180 billion in drug sales annually. I'll also be linking this article in the description below.
This is a video abstract of our paper on the pharmacogenomics of GPCR drug targets. When most people who have a disease take medication, they respond as expected and get better. However, John here does not seem to respond to this particular drug. Meanwhile, Jane seems to have developed an adverse reaction to the same medication. Why do most people respond as expected, while individuals such as John and Jane demonstrate strikingly different responses to the same medicine? One reason is the genetic differences between individuals can lead to variations in the way a person responds to a drug. Pharmacogenomics looks at how genetic differences between individuals affect and determine responses to medicine. Now, what are GPCRs and why are they important? G Protein-Coupled Receptors, or GPCRs, are membrane proteins that regulate virtually every aspect of human physiology, from the immune system to the nervous system, as well as the different sensory responses. 1/3 of all drugs in the market target GPCRs with a total sale value of $180 billion annually. They're used to treat numerous conditions such as asthma, cardiac malfunction, and diverse neurological disorders. When a drug binds to a GPCR, it can trigger or shut down diverse downstream signaling pathways inside the cell. Therefore, genetic variation in the receptors can affect how drugs bind and thus influence the effectiveness of the medicine. In our study, we asked the following questions. How variable are these GPCR drug targets in the human population? Are individuals with variant receptors likely to respond differently to drugs? What is the estimated economic burden associated with variations in GPCR drug targets? To address these questions, we analyzed multiple large-scale data sets describing the FDA-approved drugs and their targets, data on receptors in complex with diverse ligands and effectors, variant information from over 60,000 individuals, data on functional effect, and information on drug sales from the UK National Health Service. We found that on average, an individual carries 68 missense variations in approximately 1/3 of the 108 GPCR drug targets. Additionally, many FDA-approved drugs also target a number of the highly variable GPCRs. What is the likely impact of these variations? We observe that several of the variations appear within the drug binding site and other functionally important positions. For instance, 8 out of the 9 positions in the drug binding pocket of Maraviroc, an antiretroviral drug for HIV treatment, are variable in the CCR5 receptor. In this image, the green region is where the drug binds. The red regions are receptor positions that show variation in the human population. This means that patients carrying these variant receptors may show altered response when treated with this drug. The last section of our study looks at the potential economic impact of drug target variability. We developed an economic model and estimated that in the UK alone, the ineffective prescription of GPCR targeting drugs could cost the NHS between 40 million and £500 million annually. Given the extensive use of these drugs across the globe, these estimates are likely to be the tip of the iceberg in terms of global healthcare costs. To help with further research, we have also created an extensive resource within the G Protein-Coupled Receptors database. This interactive database can be found at www.gpcrdb.org. There is much more to be explored in this subject, and we hope that more research into characterizing receptor variants could increase precision of drug prescription and thereby improve quality of life for patients in the future.